Welcome to lesson 4.4. We are going to talk about protein synthesis. So far in the cell biology 2, we have looked at the structure of DNA, the structure of RNA and DNA replication. We learned that both the DNA and RNA are made up of some basic building blocks called nucleotides. But the difference is that the DNA has two long chains of these nucleotides, what we refer to as the polynucleotides, and in other words we refer to them also as the strands. So DNA is made up of two strands, whereas the RNA is made up of just one strand. Then we also talked about DNA replication, where we learned that new cells need to be formed from time to time in the body of an organism. Especially we focused on eukaryotes. So when the new cells are going to be formed, each of these new cells would need the same DNA information as existed in the original cell, and so there is a need for DNA replication to occur. In this lesson, however, we are going to look at protein synthesis to see how the body forms proteins. Before we get into what we have for this particular lesson, please remember to subscribe to our YouTube channel so that you always have our videos whenever they are released. Protein synthesis. And for our objectives, we are saying that at the end of this lesson, the student would know the importance of protein synthesis in the body. And as well, the student would be able to describe the processes involved in protein synthesis. Now, let's start by looking at what protein actually is. I know that to a lot of people, when you talk about protein, what comes to mind is food. Yes, food or nutrients. But for the sake of this topic and for science particularly, it is better for you to view protein beyond food. Protein is a large molecule made up of many amino acids. So in the body, whichever substance that has been formed by the joining of many amino acids, we refer to that particular substance as protein. So it may not always be the food that you have been thinking about. It is the chemical nature of it that we base on to refer to it as protein, right? Amino acids are compounds. So when you join so many of these amino acids, then we form the protein substance. So in the body, there are so many forms of proteins. For example, we have enzymes, hemoglobin, keratin, fibrinogen, and the rest. They are all forms of proteins. So it means that every one of these proteins were made by the joining of many amino acids. So when you look at an example like enzymes, we have so many enzymes that work in the body. Let us even consider the example of digestion. When we eat food that contains starch, the starch needs to be broken down. So the earlier steps would be breaking the starch down into maltose, and that involves an enzyme called amylase. Amylase as an enzyme was formed by the joining of many amino acids, so amylase is an example of protein. Then we also know that the red blood cells are made primarily of a substance called hemoglobin. Hemoglobin is also protein because hemoglobin was formed by the joining of many amino acids. Just like our hair and our nails are made of keratin, and keratin also is a protein substance and so was formed by the joining of many amino acids. So that is what we are going to look at, and we would say that the process by which amino acids are joined to form large protein molecules is what we term protein synthesis. So that is what we are going to look at in this particular lesson. We are going to see how many amino acids are going to be joined to form large protein molecules. Well, it is important for you to note that in the body there are about 20 different amino acids. But the point is that these amino acids may be combined differently. So depending on the kind of combination of amino acids, that is going to tell which kind of protein is going to be formed, right? So for example, I have decided to use the names like protein 1, protein 2, right? So let's look at a protein that was formed from the join of these amino acids. So like this one amino acid connected to another amino acid here, connected to another amino acid here, and connected in this sense. I have just used different colors to represent the various amino acids I'm using in this case. It doesn't mean the amino acids have these colors, right? So they have been connected. Now let's look at another protein 2 and see how the amino acids are connected. We can see here that the red amino acid, which is one of the types of amino acids that I'm using in this case, this red amino acid is connected to another green one, connected here, connected there, connected, and connected. You can see that protein 1 and protein 2 even seem to have the same kind of amino acids as their components, but the arrangements are different. And since the arrangements are different, it means that they are going to be different proteins altogether. Let's look at another example like protein 3. So also made up of this kind of combination of amino acids. So like that. So they are all going to be different proteins, although similar amino acids as their components, but the arrangements are different. So this topic is going to help us to know how the arrangement of the amino acids would be done in order to produce the protein substance that the body needs. Okay. So we'd say that the process of arranging amino acids uniquely to form a particular protein is termed protein synthesis. And note also that the process starts in the nucleus and completes in the cytoplasm. And the process is not just as straightforward as I have illustrated on the previous slide. When we move on, we are going to see the various stages involved, right? So now let's get straight into it to see the stages involved in protein synthesis. I have summarized the process in 3 stages. The stage 1 being transcription, stage 2 translation, and stage 3 packaging of the molecules into a large protein molecule. Right. So now we are going to start by looking at the stage 1, which is transcription. Well, to help you remember what transcription actually means, just have this in mind that transcription is just to say that you are copying. Right, when you're copying something, that is transcription. So what happens in the stage of transcription? Because different proteins are formed from different arrangements of the amino acids, it is important that there should be something to guide the whole process so that the arrangement of the amino acids will be done efficiently enough. So there is an information in the DNA that tells how the amino acids are supposed to be arranged to make any form of protein that the body needs. So at this stage, this is going to be copied. The information on the DNA is going to be copied by an RNA. And remember, when we were looking at the structure of RNA and its types, we talked about one type called messenger RNA. It is a messenger RNA that would copy the information on the DNA. So assuming we are going to form an enzyme like amylase, which is a protein, mRNA would copy the information in the nucleus and send it into the cytoplasm. So mRNA will copy the exact information that is needed to arrange the amino acids to form the amylase. So let us look at how it is in illustration. We know that the DNA is a helical structure. The information needed to make any protein is not found on the entire DNA molecule. It is just on a portion of it. So it is needed that that particular portion which contains the information should be unzipped. So when it is unzipped and exposed, then the mRNA can be formed against it and copy the information needed to make this particular protein. Now, there is an RNA polymerase that is responsible for the unzipping of the portion of the DNA where the information needs to be copied. So this RNA polymerase would be attached to the portion of the DNA molecule where there needs to be unzipping. And after the RNA polymerase has been attached, that portion of the DNA molecule would be unzipped. So when it has been unzipped successfully, then free nucleotides of messenger RNA, you know that in the formation of whether new DNA or new RNA, it would require nucleotides to be connected. So free nucleotides of messenger RNA would now arrange themselves against the exposed region. And as they are arranging themselves, it means they are copying the information. That is what we call transcription. So they will arrange themselves and copy the information like this. Right in here, I've just summarized it because of time. So they will copy the information. So this is mRNA formed. After mRNA has successfully copied the information against the exposed DNA, the mRNA would then be made to undergo some form of processing to make sure that the information which has been copied is right enough for the particular task ahead. So after undergoing this processing, the mRNA would leave the nucleus and enter the cytoplasm because the whole process of transcription occurs in the nucleus. So note that point. So after this, it will leave the nucleus and enter the cytoplasm. So as shown in the diagram here, it leaves into the cytoplasm. So in summary, what I've said is that portion of the DNA that contains the required information is unzipped so that free mRNA nucleotides would be arranged against the exposed DNA. And this is carried out by an enzyme called RNA polymerase. And so an mRNA is formed. The mRNA strand now leaves the nucleus into the cytoplasm so that the next stage would continue. So now we can move on to the stage 2, that is translation. So after the information has been copied by the mRNA, what happens in the next stage called translation? Let's use this diagram to understand what the process of translation is. After the messenger RNA has successfully entered the cytoplasm, the first action is that the mRNA would attach itself to a ribosome because that is a site for protein synthesis. So that is where the process of combining the amino acids actually is going to occur within the ribosome. So the mRNA would attach itself to the ribosome, and when it has attached itself, one other point for you to notice that the work of translation here is going to be between the mRNA and this structure called the tRNA. Now let me talk about the structure of the tRNA a bit. The tRNA also has nucleotides, right? But note that it has nucleotides on one end, or we would say nitrogenous bases on one end, and the other end it has an amino acid there. So you can see as shown by this particular tRNA, there are nucleotides here or nitrogenous bases here on one end, and the other end has an amino acid. You can see a similar thing here. The nucleotides here, the amino acid here. Interestingly, the tRNA has the nucleotides or the nitrogenous bases appearing as triplets. Right. 1, 2, 3. 1, 2, 3. 1, 2, 3. And just like you see that this particular tRNA has this code, this triplet code at its one end. And that triplet code corresponds to this particular amino acid. You see a similar thing here. This one also has this code. So you see CAGU here having this amino acid at its other end. And you see ACG here having this particular amino acid also at its end. What does it tell us? It means that every triplet code has a unique amino acid that it carries. So let's note that point. Every triplet code has its own amino acid that it carries. Right. So the work is going to be this way. Since the tRNA always comes with a triplet code, what we refer to as anticodon, it would be arranged against the messenger RNA also in threes. So you can see here that this is the tRNA. tRNA has 1, 2, 3 here, and it has paired with the mRNA also in threes like this. So in threes here, in threes, like that. Since the mRNA came with the original code, we call it the codon, right? And then the tRNA is going to form against the mRNA, but in the opposite way, because you know that the pairing rule, guanine will pair with cytosine, cytosine here pairing with guanine, adenine pairing with uracil. So the pairing is going to be in the opposite way. So if the mRNA has every three or triplet code being referred to as a codon, then the tRNA coming the opposite way with its code is going to be referred to as the anticodon. So the anticodons on the tRNA would be paired against the codons on the mRNA. So in threes, this one, that. So like that, in threes. Now, as the tRNA keeps arranging its anticodon against the messenger RNA's codons, at the same time, the amino acids that are connected on the other end of the tRNAs would also be connecting themselves. That is so beautiful. We are arranging ourselves with our anticodon, and as we are arranging ourselves with our anticodon, at the same time, the amino acids are also arranging themselves on the other end. So by the time the tRNAs have been successfully arranged against the mRNA, against the entire mRNA length, by that time, the amino acids have also been correctly connected to form a long chain of amino acids, what we refer to as the polypeptide chain. So like this. And the amino acids are going to be connected by what we call the peptide bonds. Right. So in summary, I would say that during the translation, messenger RNA attaches itself to a ribosome. Then tRNA with its anticodon would arrange themselves against the messenger RNA. And as they arrange themselves, the amino acids at the other end of the tRNA would also connect themselves to form a polypeptide chain. And after that has been successfully done, then the process of translation is complete. Let's take a summary of what I've talked about under translation. So I said, the tRNA picks up amino acids with its specific anticodon 3 nucleotides. Then, as the mRNA is now in the cytoplasm, the tRNA would be arranged along the mRNA strand. So the anticodons on the tRNA pairs with the codons on the mRNA. But meanwhile, the amino acids on the tRNA also join to form a large molecule of many amino acids that we call polypeptide. So that is the process of translation for the stage 2. Well, note also that in our lesson on structure and types of RNA, we mentioned ribosomal RNA, but we haven't talked about the function of the ribosomal RNA. Well, its function is to direct the catalytic steps involved in protein synthesis. So that means the steps involved in the joining of the many amino acids. Directing those steps, that is what the rRNA does. That is the ribosomal RNA. And you can even guess from the name ribosomal RNA. Finally, let's go to packaging, which is the final stage in many cases, but also a very simple stage. The polypeptide formed may join with other polypeptides to form a large protein. In many instances, it is realized that after the amino acids have been joined to form the long polypeptide chain, that polypeptide chain may not be enough to be a functional protein in the body. And so, it would have to be joined to other polypeptides which have also been formed within the cell. And when they have been joined successfully, then they will form a large protein molecule. Because one polypeptide chain, another one, joining themselves together, then they become a very large protein. Right. In some cases too, apart from the polypeptide chains joining, there is a need for the body to also add several other substances, so like lipids or carbohydrates, so that they will package the protein into its functional form. So when the body has done all of these, then we would say that the whole process of protein synthesis is now complete. Yes. So that will be the end of protein synthesis. So far, I have talked about protein synthesis, which could be summarized in these main stages. I talked about transcription. That is the stage of copying where mRNA would copy information against the exposed DNA, and it occurs only on a part of the DNA. Then there is translation, where the information that was copied by the mRNA would have to be copied by the tRNA, arranging itself against the mRNA. And as it is arranging itself, on the other end too, the amino acids will be connecting to form the long polypeptide chain that would result in the formation of the protein. So in many cases, finally, there would be packaging of the many polypeptide chains into large protein molecule. Right. Just like I always do, before we end our lesson for today, please, I have a few questions for you to try them. So the first question is that in protein synthesis, I would like you to state what happens during transcription. And let me also know your thoughts on what happens during translation. The second question is, list 4 examples of proteins formed in the body. That's very simple and straightforward. Then finally, name one enzyme that is involved in transcription. Okay, thank you for being part of our lesson today. Please don't forget to subscribe to our YouTube channel so that you always have our videos whenever they are released. See you in our next class.